Picture Earth 4 billion years ago. No blue skies stretch overhead instead. An orange haze of methane and ammonia shrouds the planet. The surface burns at temperatures that would melt lead. Volcanic eruptions tear through the crust every few hours, spewing molten rock into an atmosphere so toxic it would kill any modern life form in seconds. Meteors the size of mountains slam into the planet daily, each impact releasing energy equivalent to billions of nuclear bombs. This is not a world that seems capable of asking questions about itself. Yet somewhere in this hellish landscape, something extraordinary begins to happen. In shallow pools warmed by a younger, dimmer sun, molecules start behaving differently. They begin copying themselves. They start competing. They develop the ability to change and improve. Without knowing it, they begin the greatest experiment in the history of the universe, the experiment we call life. Here's what's really happening in those primordial pools. Water molecules dance with carbon, nitrogen, and phosphorus in combinations that shouldn't be stable. But occasionally, just occasionally, they form structures that can make copies of themselves. These aren't living things as we understand them. They're more like chemical recipes that accidentally learned to replicate. Each successful copy marks a small victory against the universe's tendency toward chaos. The universe, you see, prefers disorder. Energy spreads out, heat dissipates, and complex structures fall apart. This principle, called entropy, governs everything from cooling coffee to dying stars. But in those ancient tidal pools, something begins pushing back against entropy. Simple molecules organize into more complex patterns. They start using energy from their environment to maintain their structure and make copies. They become the first rebels in the cosmic story, tiny pockets of increasing order in a universe trending toward chaos. We exist in a time when asking questions about life feels natural, even inevitable. But for most of Earth's history, there was no one here to wonder about anything. No consciousness pondered the meaning of existence. No minds marveled at the complexity of living systems. The questions we now ask about life had to wait nearly 4 billion years for questioners to evolve. The first humans to systematically wonder about life weren't scientists in the modern sense. They were shamans, healers, and observers who noticed patterns in the natural world. They watched caribou migrate across frozen landscapes, following invisible pathways carved into their genetic memory. They observed salmon swimming upstream against impossible currents, driven by instincts older than mountains. They saw birds navigate across thousands of miles of open ocean, guided by magnetic fields they couldn't see but somehow sensed. These early observers developed the first theories about what makes something alive. They noticed that living things moved with purpose, that they responded to their environment, that they could heal from injuries and produce offspring. Most importantly, they realized that when life left a body, something fundamental changed. The same carbon, oxygen, and nitrogen remained, but the spark that animated them vanished. In ancient Greece, a philosopher named Aristotle took these observations and made them systematic. Around 350 years before the Common Era, he began cataloguing every living thing he could find. His students brought him specimens from across the Mediterranean, fish from the Aegean Sea, birds from Egyptian marshes, insects from Macedonian forests. Aristotle examined each one with meticulous care, documenting their structures, behaviors, and relationships to other organisms. Aristotle's work revealed something remarkable. Life follows patterns. Animals with backbones share similar body plans. Creatures that lay eggs tend to have certain characteristics. Plants that produce flowers reproduce differently from those that don't. He identified 354 different animal species and developed the first systematic classification of living things, a project that wouldn't be completed until 23 centuries later. But Aristotle made an even more profound observation. He noticed that all living things seem to have an inherent purpose, a built-in drive toward their own perfection. A seed doesn't just randomly change. It develops into exactly the kind of plant encoded in its structure. An embryo doesn't grow into any random shape. It becomes precisely the animal it was destined to be. This suggested that life operated according to principles different from the simple mechanical forces that governed rocks and water. For the next 2,000 years, this view dominated human thinking about life. Living things were fundamentally different from non-living matter. They possessed a vital force, an essence that couldn't be reduced to mere chemistry and physics. Life was animated by something beyond the material world, a spark that transformed dead matter into living beings. This understanding began to change in the 1600s, when a Dutch fabric merchant named Antony van Leeuwenhoek started grinding lenses as a hobby. Van Leeuwenhoek wasn't trained as a scientist. He had no formal education beyond elementary school. He simply possessed an obsessive curiosity about the world around him and an extraordinary talent for making precise optical instruments. In 1674, van Leeuwenhoek created a microscope more powerful than any that had existed before. His lenses could magnify objects nearly 300 times, revealing details invisible to the naked eye. He began examining everything he could find: samples of pond water, scrapings from his own teeth, drops of blood, fragments of plants and insects. What he discovered changed everything we thought we knew about life. On an autumn morning in 1676, van Leeuwenhoek placed a drop of rainwater under his microscope and peered through the lens. What he saw made him gasp. The water teemed with tiny creatures, each one moving with obvious purpose. They swam, they spun, they chased each other through the liquid landscape. Some looked like miniature eels, others resembled tiny spheres covered in waving hairs. All of them were clearly, unmistakably alive. Van Leeuwenhoek called these creatures animalcules, little animals. He had discovered a hidden universe that existed parallel to our own, populated by beings so small that millions could live in a single drop of water. Every puddle, every pond, every moist surface harbored vast communities of microscopic life. Here's what van Leeuwenhoek couldn't have known. Those tiny creatures he observed were actually the most successful life forms on Earth. Bacteria and other microorganisms make up more than half of all living matter on the planet. They exist in environments that would kill any large organism in boiling hot springs, in highly acidic pools, in the deepest ocean trenches, even in solid rock miles beneath the surface. They've been evolving for over 3.5 billion years, developing biochemical capabilities that dwarf anything larger organisms can accomplish. A few years later, in 1678, the English scientist Robert Hooke published his own microscopic observations. Hooke had been examining thin slices of cork under his microscope when he noticed something extraordinary. The cork wasn't a solid mass, as it appeared to the naked eye. Instead, it consisted of countless tiny chambers, each one separated from its neighbours by thin walls. The structure reminded Hooke of the small rooms where monks lived in monasteries, chambers they called cells. Hooke coined the term cell to describe these microscopic compartments, unknowingly giving a name to the basic unit of all life. What he was seeing in that piece of cork were the remnants of once living plant cells, their walls still intact long after the cellular contents had dried up and disappeared. The discovery of cells revealed life's fundamental secret. Every living thing, from the smallest bacterium to the largest whale, consists of these microscopic chambers. Some organisms, like bacteria, consist of just a single cell. Others, like humans, contain approximately 37 trillion cells, each one a miniature factory performing the chemistry that keeps us alive. Inside each cell, chemical reactions occur that would make any industrial chemist envious. Proteins fold into precise three-dimensional shapes that allow them to catalyze specific reactions. DNA stores information in a 4-letter code more efficient than any computer system humans have devised. Molecular machines transport materials, generate energy, and construct new cellular components with precision that exceeds our most advanced manufacturing processes. But perhaps the most remarkable thing about cells is their ability to reproduce. When a cell divides, it creates an exact copy of itself, complete with all the molecular machinery needed to sustain life. This process requires the cell to duplicate its entire DNA library, typically millions of genetic letters, and distribute perfect copies to each daughter cell. The precision required is extraordinary. A single mistake in copying might prove fatal. As van Leeuwenhoek and his contemporaries peered deeper into the microscopic world, they began to realize that the boundary between living and non-living wasn't as clear as it had seemed. The vital force that supposedly animated living matter appeared to operate through the same chemical and physical principles that governed the rest of the universe. Life wasn't separate from the material world. It was an extraordinarily complex expression of it. This realization sparked a question that still drives scientific inquiry today. If living things operate according to the same natural laws as everything else, what makes them special? What distinguishes a living cell from a sophisticated machine? The answer lies in something called emergence. Just as individual notes combine to create music, or individual pixels combine to create images, the molecules in living systems interact in ways that produce properties greater than the sum of their parts. Life emerges from chemistry, but it's chemistry organized in patterns of staggering complexity and elegance. Consider what happens when you digest food. Your stomach doesn't simply dissolve nutrients, it orchestrates a symphony of chemical reactions. Enzymes break down proteins into amino acids, carbohydrates into sugars, and fats into fatty acids. These molecules travel through your bloodstream to cells throughout your body, where other enzymes reassemble them into the specific proteins, carbohydrates, and fats your body needs. The whole process requires thousands of different chemical reactions, each one precisely controlled and coordinated with all the others. Here's what makes this truly remarkable. Every atom involved in this process obeys exactly the same physical laws that govern rocks, water, and stars. Carbon atoms don't behave differently inside living things than they do in diamonds or graphite. Oxygen molecules follow the same rules whether they're in the atmosphere or in your bloodstream. Yet when these atoms organize into the patterns we call life, they collectively exhibit behaviors, growth, reproduction, adaptation, consciousness that seem almost magical. The questions that first stirred in those ancient tidal pools have evolved into the deepest mysteries humans have ever contemplated. How did non-living matter first organize itself into living systems? How do billions of individual cells coordinate to create a thinking, feeling organism? How did evolution produce brains capable of understanding their own existence? These questions connect us directly to those early dutting observers who watched migrating herds and wondered what drove them. They link us to Aristotle cataloguing species in ancient Greece, to van Leeuwenhoek marveling at his microscopic menagerie, to every human who has ever looked at the natural world and asked, how does this work? The story we're about to explore, the story of life on Earth, is really the story of how the universe learned to ask questions about itself. From those first self-replicating molecules in primordial pools to the minds now reading these words, life represents something unprecedented in cosmic history. Matter organized in such a way that it can contemplate its own existence. We stand at a unique moment in this 4 billion year story. For the first time, living beings can trace their own origins back to the very beginning. We can peer into cells and see the molecular machinery that makes life possible. We can read the genetic code that connects us to every other living thing. We can even create artificial versions of the processes that sparked life in the first place. But every answer we discover reveals new questions. The more we learn about life's mechanisms, the more remarkable they appear. The more we understand about life's history, the more improbable its emergence seems. The more we explore the cosmos, the more mysterious our own existence becomes. That's the paradox at the heart of life's story. The better we get at answering questions about life, the more questions we discover. Each revelation opens new frontiers of mystery. Each solved puzzle reveals 10 more puzzles we never knew existed. The journey we're about to take will carry us from those first molecular rebels in ancient seas to the most sophisticated life forms the universe has ever produced. We'll witness the emergence of cells, the invention of photosynthesis, the development of complex organisms, and the evolution of consciousness itself. We'll explore how life has survived 5 mass extinctions, how it has colonized every environment on Earth, and how it might exist on other worlds. Most importantly, we'll discover how the questions that first stirred in primordial pools have evolved into the scientific methods, technological capabilities, and philosophical insights that now allow us to understand our place in the cosmic story. The universe spent 13 8/10 billion years preparing for the emergence of life on Earth. In the next chapter, we'll explore how that life learned to see itself for the first time, opening doorways to hidden worlds that exist all around us, waiting to be discovered. On a crisp morning in 1676, Antony van Leeuwenhoek walked to his favourite pond near Delft, Holland. He carried with him a small brass microscope, no bigger than his palm, fitted with a lens he had ground himself using techniques he never revealed to anyone. What happened next would change humanity's understanding of life forever. Van Leeuwenhoek placed a single drop of pond water between 2 thin pieces of glass and positioned the sample beneath his lens. As he peered through the microscope and adjusted the focus, his eyes widened in amazement. The water wasn't empty, it was alive with countless tiny creatures, each one swimming, spinning, and darting through the liquid with obvious purpose. I now saw very plainly that these were little eels or worms, lying all huddled up together and wriggling, Van Leeuwenhoek wrote in his journal. This was for me, among all the marvels that I have discovered in nature, the most marvellous of all. What Van Leeuwenhoek had discovered was the microscopic world, a hidden universe that exists parallel to our own, populated by creatures so small that millions can live in a space smaller than the period at the end of this sentence. These animalcules, as he called them, revealed life's most fundamental secret. Everything living, from the smallest bacteria to the largest whale, is built from microscopic units, called cells. Here's what makes this discovery so extraordinary. Van Leeuwenhoek's microscope could magnify objects nearly 300 times, revealing details completely invisible to the naked eye. At this magnification, a typical bacterial cell appears about the size of a grain of rice, but even this tiny scale doesn't capture the true magnitude of what he had found. A single drop of pond water contains more living organisms than there are people on Earth. These microscopic creatures weren't just randomly moving, they displayed complex behaviors that suggested sophisticated internal machinery. Some spun like tiny corkscrews, propelling themselves through the water with remarkable efficiency. Others extended long, whip-like appendages that beat in coordinated waves, pushing them forward like microscopic swimmers. Many changed direction suddenly, as if responding to invisible signals in their environment. Van Leeuwenhoek spent the next 50 years documenting this hidden world. He examined water from rivers, lakes, and rain puddles. He studied samples from his own mouth, discovering that the number of living creatures in the scurf of a man's teeth are so many that I believe they exceed the number of men in a kingdom. He observed bacteria from his intestines, protozoans from flower vases, and microorganisms from every imaginable source. What emerged from these decades of careful observation was a revolutionary understanding. Life exists at scales far beyond what our senses can detect. The visible world we inhabit represents only the tip of an enormous biological iceberg. Beneath our perception lies a vast ecosystem of microscopic organisms that outnumber, outweigh, and in many ways outperform all larger forms of life. But Van Leeuwenhoek's discoveries raised more questions than they answered. What were these tiny creatures made of? How did they move and reproduce? Most puzzling of all, how could something so small be alive in the same way as plants, animals, and humans? The answers began emerging through the work of Robert Hooke, an English scientist who approached the microscopic world from a different angle. While van Leeuwenhoek focused on living organisms, Hooke examined dead materials, thin slices of cork, pieces of wood, fragments of charcoal. In 1665, he published Micrographia, a book filled with detailed drawings of objects viewed under magnification. When Hooke examined cork under his microscope, he made a startling discovery. The cork wasn't solid as it appeared to the naked eye. Instead, it consisted of countless tiny chambers, each one separated from its neighbors by thin walls. The structure reminded him of the small rooms in monasteries, where monks lived, rooms called cells. Hooke wrote, "These pores, or cells, were not very deep, but consisted of a great many little boxes." Hooke had unknowingly discovered the fundamental building blocks of life. What he was seeing in that piece of cork were the empty shells of plant cells, their walls still intact long after the living contents had dried up and disappeared. Every living thing on Earth, from microscopic bacteria to massive sequoia trees, is constructed from these cellular units. The implications of this discovery were staggering. If all life consists of cells, then understanding how cells work would unlock the secrets of life itself. Over the next 2 centuries, scientists began peering deeper into cellular structure, revealing layers of complexity that exceeded anything they had imagined. Inside each cell, they discovered a bustling metropolis of molecular activity. The cell membrane acts like a sophisticated border control system, deciding which molecules can enter and exit. The nucleus serves as a command center, containing the DNA blueprints that direct all cellular activities. Mitochondria function as power plants, converting nutrients into usable energy. Ribosomes operate as protein factories, assembling complex molecules according to genetic instructions. Here's what makes cellular organization truly remarkable. Every process happens with extraordinary precision, despite the chaotic environment of molecular motion. Inside a typical human cell, molecules move at speeds of hundreds of meters per second, colliding billions of times each second in what should be complete pandemonium. Yet somehow, the right molecules find each other at exactly the right time to carry out precisely the reactions needed to sustain life. Consider what happens when a cell needs to make a specific protein. The process begins in the nucleus, where enzymes unzip the DNA double helix and copy the genetic instructions for that protein onto a molecule of messenger RNA. This RNA travels out of the nucleus to a ribosome, where it serves as a template for protein construction. Transfer RNA molecules bring the correct amino acids to the ribosome in the precise order specified by the genetic code. The ribosome links these amino acids together, creating a protein that folds into exactly the 3-dimensional shape needed to perform its function. This entire process happens thousands of times per second in every cell of your body. Each step requires dozens of different enzymes and molecular machines, all working in perfect coordination. The precision required is extraordinary. A single mistake in the sequence could produce a protein that doesn't work, potentially harming or killing the cell. But cells don't just maintain themselves, they also reproduce. Cell division represents one of the most remarkable processes in nature. When a cell prepares to divide, it must first duplicate its entire DNA library, typically containing millions of genetic letters. This duplication happens with incredible accuracy, less than 1 error per billion letters copied. The cell then organizes its duplicated chromosomes and distributes them equally between 2 daughter cells. Each daughter cell receives a complete copy of the genetic information, along with all the molecular machinery needed to sustain life. The whole process transforms one living cell into 2 identical living cells, each capable of carrying on the full range of life's activities. As scientists developed more powerful microscopes, they discovered that cells come in two fundamentally different types. Prokaryotic cells, found in bacteria and archaea, have relatively simple internal organization. Their genetic material floats freely in the cell without being enclosed in a nucleus. These cells represent life's original design, the cellular architecture that dominated Earth for nearly 2 billion years. Eukaryotic cells, found in plants, animals, fungi, and many microorganisms, display much more complex internal organization. Their genetic material is enclosed in a membrane-bound nucleus, and their interiors contain numerous specialized compartments called organelles. These cells represent a later evolutionary innovation that made possible the development of complex multicellular organisms. The difference between these cell types reveals something profound about life's history. Prokaryotic cells evolved first, developing all the basic biochemical processes that sustain life. Eukaryotic cells emerged later through a process that scientists are still working to understand, a process that involved one cell engulfing another and forming a permanent partnership. Here's what we know about this remarkable transformation. Around 2 billion years ago, certain bacteria evolved the ability to use oxygen for energy production, a process called aerobic respiration that generates far more energy than any previous metabolic system. Other cells began engulfing these oxygen-using bacteria, initially perhaps as food, but instead of digesting them, some host cells kept the bacteria alive inside their own cellular boundaries. This arrangement benefited both partners. The engulfed bacteria received protection and nutrients from their hosts. In return, they provided their hosts with abundant energy from aerobic respiration. Over millions of years, these bacterial partners became permanently integrated into their host cells, evolving into the organelles we now call mitochondria. A similar process occurred with photosynthetic bacteria, which became the chloroplasts found in plant cells. These evolutionary partnerships, called endosymbiosis, represent some of the most important events in life's history. They made possible the development of complex cells with far greater energy resources than any prokaryotic cell could achieve alone. The discovery of cellular structure also revealed why life can exist at all in a universe governed by entropy. Individual molecules follow the laws of thermodynamics. They tend toward disorder, their energy dissipates, and their structures eventually break down. But cells create islands of order by constantly consuming energy and using it to maintain their organization. Think of a cell as a sophisticated eddy in a stream. The water molecules in an eddy move in organized patterns that seem to defy the overall flow of the stream. But the eddy exists only because energy from the flowing water constantly maintains its structure. Remove the energy source, and the eddy immediately disappears back into random flow. Similarly, cells maintain their organization by constantly consuming energy from their environment. They break down nutrients, capture energy from sunlight, or harvest energy from chemical reactions. This energy powers the molecular machines that maintain cellular structure, repair damage, and carry out all the processes we associate with life. When the energy stops flowing, the cell dies, and its organized structures rapidly break down into simpler components. The same carbon, oxygen, nitrogen, and other atoms remain, but they lose the precise organization that enabled them to function as a living system. The most dramatic cellular discovery came in 1953, when Rosalind Franklin's X-ray crystallography revealed the structure of DNA. Franklin's photographs showed that DNA molecules form a double helix, two intertwined spirals held together by chemical bonds between complementary base pairs. This structure immediately suggested how genetic information could be stored and copied. James Watson and Francis Crick used Franklin's data to build the first accurate model of DNA structure. Their model revealed that DNA consists of two complementary strands, each one a sequence of 4 different chemical letters: adenine, guanine, cytosine, and thymine. These letters pair in specific combinations. Adenine always pairs with thymine, guanine always pairs with cytosine. This pairing rule means that each strand of DNA contains the information needed to reconstruct its partner. When a cell prepares to divide, enzymes unzip the double helix and use each strand as a template to build a new complementary strand. The result is two identical DNA molecules, each containing the same genetic information as the original. Here's what makes DNA structure so elegant. It solves the fundamental problem of how information can be copied accurately across generations. The base pairing rules ensure that errors in copying are rare, while the double-stranded structure provides redundancy. If one strand is damaged, the other can serve as a backup copy for repairs. But DNA does more than just store information. It contains the instructions for building and operating every cell. The sequence of chemical letters in DNA determines which proteins a cell can make, how it responds to its environment, when it divides, and ultimately what type of organism it becomes. A typical human cell contains 23 pairs of chromosomes, each one a long DNA molecule containing millions of genetic letters. If you could unwind all the DNA in a single human cell and stretch it out in a straight line, it would extend about 6 feet. Yet, this enormous amount of information is packed into a nucleus, only about 5 micrometers across, smaller than most bacteria. The information density in DNA exceeds any data storage system humans have devised. The complete human genome contains approximately 3.2 billion letters of genetic code, enough information to fill about 5,000 books. Yet all this information fits into a space millions of times smaller than the head of a pin. Modern microscopy has revealed even more extraordinary details about cellular organization. Electron microscopes can magnify objects up to 2 million times, revealing the intricate architecture of molecular machines inside cells. These images show ribosomes assembling proteins, mitochondria generating energy, and chromosomes organizing for cell division with mechanical precision that rivals any human engineering. Perhaps most remarkably, we now know that the molecular machines in cells operate according to the same physical principles that govern larger mechanical systems. DNA polymerase moves along DNA strands like a microscopic train on molecular tracks. ATP synthase rotates like a turbine to generate cellular energy. Kinesin motors walk along cellular highways, carrying cargo to specific destinations. These molecular machines demonstrate that life operates as a form of nanotechnology, biological systems that manipulate matter at the atomic and molecular scale with extraordinary precision. But unlike human technology, biological nanotechnology is self-assembling, self-repairing, and capable of reproduction. The hidden universe of cells that Van Leeuwenhoek first glimpsed has turned out to be far more complex and remarkable than he could have imagined. Every cell represents a complete living system, carrying out thousands of precisely coordinated chemical reactions while maintaining its organization in the face of constant molecular chaos. Understanding cellular structure and function has revolutionized our profound comprehension of life itself. We now know that all the properties we associate with living things, growth, reproduction, response to environment, metabolism, emerge from the organized activities of cells. Life isn't animated by a mysterious vital force. It's the result of extraordinary molecular organization operating according to the same natural laws that govern everything else in the universe. But this understanding has also deepened the mystery of life's origins. If cells are so complex, requiring thousands of different molecular components working in perfect coordination, how did the first cells ever arise from non-living matter? How did random chemical reactions in primordial environments give rise to the sophisticated molecular machinery we see in every living cell? These questions lead us to the next chapter in life's story, the search for life's ultimate beginnings in Earth's violent youth, where chemistry first learned to rebel against entropy and create the organized complexity we call life. Imagine Earth 3 8/10 billion years ago. The planet spins through space as a hellish ball of molten rock and toxic gases. Surface temperatures soar above 1,200 degrees Celsius, hot enough to melt copper. The atmosphere contains no oxygen, only clouds of methane, ammonia, and hydrogen sulfide that would be instantly lethal to any modern life form. Volcanic eruptions tear through the crust every few hours, spewing lava and ash into skies that never see sunlight through the orange haze. Mountain-sized asteroids slam into the planet daily, each impact releasing enough energy to vaporize entire oceans. The young moon looms much closer than today, raising tides hundreds of feet high that sweep across the continents twice each day. Lightning storms rage continuously, striking the surface millions of times per hour with electrical discharges that dwarf anything we see today. This is the world where life began. Here's what makes this so extraordinary. In this landscape that seems designed to destroy any form of organization, the universe's most complex and improbable phenomenon somehow emerged. Against all odds, simple molecules learn to copy themselves, compete for resources, and evolve into increasingly sophisticated forms. The first spark of life ignited in conditions that would annihilate any living thing on Earth today. Scientists have spent decades trying to understand how this miracle occurred. How did non-living chemicals organize themselves into the first living systems? What specific conditions allowed simple molecules to cross the threshold from chemistry to biology? Most fundamentally, how did the universe's tendency toward disorder give birth to life's extraordinary complexity? The search for answers began in 1952, when a graduate student named Stanley Miller decided to recreate primordial Earth in his laboratory. Working with his advisor Harold Urey at the University of Chicago, Miller constructed an apparatus that simulated the conditions scientists believed existed on early Earth. Miller's experimental setup was elegantly simple. He filled a flask with water to represent the primordial oceans. Above the water, he created an atmosphere of methane, ammonia, and hydrogen, the gases scientists thought dominated early Earth's atmosphere. He then subjected this mixture to electrical sparks that simulated the constant lightning storms that raged across the young planet. For a week, Miller's artificial lightning crackled through his synthetic atmosphere, while heated water vapor circulated through the system like an accelerated water cycle. He expected interesting chemistry to occur, but what happened exceeded his wildest expectations. Within just a few days, the clear water in Miller's apparatus began turning reddish-brown. Chemical analysis revealed that the mixture now contained amino acids, the building blocks of proteins. These weren't just random organic molecules, but specifically the same amino acids that form the basis of all life on Earth. In a single week, Miller had recreated processes that might have taken millions of years on the early Earth. Here's what was happening inside Miller's apparatus at the molecular level. The electrical sparks provided energy that broke apart the simple molecules in the atmosphere. Methane split into carbon and hydrogen fragments. Ammonia broke down into nitrogen and hydrogen components. Water molecules separated into hydrogen and oxygen atoms. These molecular fragments then recombined in new ways, forming increasingly complex organic compounds. The amino acids that formed in Miller's experiment weren't just any organic molecules, they were precisely the ones used by all living things today. Glycine, alanine, aspartic acid, and glutamic acid appeared in his flask, along with several others that play crucial roles in biological systems. The experiment demonstrated that the chemical building blocks of life could form spontaneously under conditions that likely existed on early Earth. But Miller's experiment also revealed a deeper principle about how complexity emerges from simplicity. When energy flows through a system containing simple molecules, those molecules don't just break apart randomly, they organize into increasingly complex patterns. Energy drives the formation of larger, more sophisticated molecular structures that couldn't exist in isolation. Over the decades following Miller's breakthrough, scientists have recreated many other aspects of early Earth chemistry. They've shown that simple organic molecules can form in hydrothermal vents, in the atmospheres of gas giant planets, and even in the vacuum of space. Amino acids have been found in meteorites, suggesting that some of life's building blocks might have arrived from outer space during Earth's heavy bombardment period. But amino acids are just the beginning of life's story. To create actual living systems, these building blocks must organize into functional proteins, nucleic acids, and cellular membranes. This requires not just the right chemical ingredients, but also the right conditions for them to assemble into working biological machinery. The most promising location for this assembly process appears to be hydrothermal vents on the ocean floor. These underwater geysers spew superheated water at temperatures reaching 360 degrees Celsius, creating chemical conditions unlike anywhere else on Earth's surface. Here's what makes hydrothermal vents so special for the origin of life. The vents create strong temperature gradients, with water temperatures dropping from over 300 degrees to near freezing within just a few centimeters. These gradients drive chemical reactions that wouldn't occur under uniform conditions. The vents also provide a steady supply of minerals and dissolved gases that can serve as raw materials for organic chemistry. Most importantly, hydrothermal vents create natural chambers and surfaces where molecules can concentrate and organize. The rocky chimneys around vents contain countless tiny pores and cavities that can trap organic molecules and allow them to reach high concentrations. These natural laboratories might have provided the protected environments where the first self-replicating systems evolved. Scientists have discovered that the chemistry around modern hydrothermal vents produces many of the same organic compounds found in living systems. These include not just amino acids, but also nucleotide bases that form the building blocks of RNA and DNA. Even more remarkably, some of the chemical reactions around vents can produce simple proteins and short stretches of RNA without any biological enzymes. But the most crucial discovery about hydrothermal vents concerns energy flow. Life requires a constant supply of energy to maintain its organization and carry out its functions. On modern Earth, most life ultimately depends on energy from the sun, captured through photosynthesis. But photosynthesis is a complex process that couldn't have existed in the earliest life forms. The chemistry around hydrothermal vents provides an alternative energy source that doesn't require sunlight. Chemical reactions between hot vent fluids and seawater can generate the same type of energy that modern cells use to power their metabolism. This suggests that the first life forms might have been chemosynthetic organisms that derived energy from chemical reactions rather than from sunlight. The discovery of extremophile organisms living around modern hydrothermal vents supports this hypothesis. These remarkable creatures thrive in conditions that would be instantly lethal to most life forms in water, hot enough to cook an egg, in chemical environments more acidic than battery acid, under pressures that would crush a human instantly. Yet they not only survive but flourish, forming the basis of entire ecosystems that exist in complete darkness. Some of these extremophiles use metabolic processes that might be similar to those of the earliest life forms. They derive energy from hydrogen sulfide, methane, and other chemicals that were abundant on early Earth. Their cellular machinery operates efficiently at high temperatures and in highly acidic conditions. Most remarkably, their biochemistry appears simpler and more primitive than that of organisms living in more moderate environments. These discoveries have led scientists to propose a new model for life's origins called the metabolism-first hypothesis. According to this idea, the first step toward life wasn't the formation of self-replicating molecules like RNA or DNA. Instead, life began with the establishment of metabolic networks, sets of chemical reactions that could sustain themselves and grow more complex over time. Here's how this process might have worked. In the chemical environment around hydrothermal vents, certain combinations of reactions naturally reinforce each other. The products of one reaction serve as the raw materials for another reaction, which produces compounds that catalyze the first reaction. These self-sustaining chemical networks can grow and become more complex without requiring any genetic information or replication machinery. Over time, these metabolic networks might have incorporated molecules that could store information and catalyze reactions more efficiently. RNA molecules are particularly interesting in this context because they can both store genetic information and catalyze chemical reactions. This dual functionality makes RNA a candidate for bridging the gap between simple chemistry and true biology. The RNA world hypothesis suggests that early life was based on RNA rather than DNA. RNA molecules can fold into complex three-dimensional shapes that allow them to catalyze chemical reactions, much like protein enzymes do in modern cells. But unlike proteins, RNA molecules can also store genetic information and make copies of themselves. Scientists have demonstrated that RNA molecules can evolve in laboratory conditions. When researchers create populations of RNA molecules and subject them to selection pressure, the molecules mutate and adapt just like living organisms. Some RNA molecules become better at catalyzing specific reactions, others become more efficient at replication, and still others develop new functions entirely. This experimental evolution of RNA suggests that the transition from chemistry to biology might have occurred gradually rather than in a single dramatic event. Simple self-replicating RNA molecules could have emerged from the chemical soup around hydrothermal vents, then evolved increasing complexity through natural selection operating at the molecular level. But RNA-based life faces significant challenges. RNA molecules are chemically unstable, breaking down rapidly in water. They're also less efficient than proteins at catalyzing most chemical reactions, and less stable than DNA for storing genetic information. For these reasons, scientists believe that RNA-based life was eventually replaced by the DNA-protein systems that dominate modern biology. The transition from RNA to DNA-protein life represents one of the most important events in biological history. DNA is much more stable than RNA, making it better suited for long-term information storage. Proteins are more versatile catalysts than RNA, allowing for more sophisticated biochemistry. But this transition required the evolution of complex molecular machinery that could translate genetic information from DNA into functional proteins. This machinery, including ribosomes, transfer RNAs, and numerous enzymes, is extraordinarily complex. The ribosome alone contains over 50 different proteins and several RNA molecules, all working together with mechanical precision to manufacture proteins according to genetic specifications. How such sophisticated molecular machinery could have evolved from simple RNA-based systems remains one of the greatest mysteries in biology. Recent discoveries about the ribosome's structure provide important clues. The core of the ribosome, the part that actually links amino acids together to form proteins, consists entirely of RNA. The protein components appear to have been added later in evolution, suggesting that ribosomes evolved from simpler RNA-based catalysts. This finding supports the idea that the transition from the RNA world to modern biology occurred through a series of gradual improvements rather than through sudden revolutionary changes. Each improvement provided some advantage that helped organisms compete more effectively for resources or reproduce more efficiently. The emergence of cellular membranes represents another crucial step in life's early evolution. Membranes are essential for life because they create boundaries that separate living systems from their environments. Without membranes, the complex chemistry of life would simply diffuse away into the surrounding medium. Scientists have shown that simple membranes can form spontaneously under certain conditions. When certain organic molecules called amphiphiles are mixed with water, they automatically organize into membrane-like structures. These molecules have one end that attracts water and another end that repels it, causing them to arrange themselves into sheets with their water-loving ends facing outward. These spontaneous membranes can even form enclosed spheres that resemble primitive cells. Under the right conditions, these membrane spheres can grow, divide, and encapsulate organic molecules from their surroundings. While they're not alive in any meaningful sense, they demonstrate how some of the basic features of cellular organization could have emerged from simple physical processes. The combination of self-replicating RNA, metabolic networks, and membrane boundaries might have produced the first true living cells. These primitive organisms would have been far simpler than any modern life form, but they would have possessed the fundamental properties that define life: the ability to metabolize, reproduce, and evolve. Once this threshold was crossed, evolution could begin operating to increase complexity and efficiency. Organisms that could reproduce faster, metabolize more efficiently, or survive in new environments would have advantages over their competitors. Over millions of years, these advantages would accumulate, leading to increasingly sophisticated forms of life. The fossil record provides tantalizing glimpses of this early evolution. The oldest fossils that might represent living organisms are approximately 3.5 billion years old structures called stromatolites that appear to be formed by ancient bacterial communities. These fossils suggest that by this time, life had already evolved complex behaviors like photosynthesis and had begun forming multicellular communities. But the gap between the emergence of the first self-replicating systems and these complex fossil organisms spans several 100 million years. During this crucial period, life evolved from simple chemical networks into sophisticated cellular organisms capable of photosynthesis, complex metabolism, and coordinated group behavior. Understanding this transition remains one of the greatest challenges in biology. How did simple self-replicating molecules evolve into cells with hundreds of different proteins working in precise coordination? How did random chemical reactions give rise to the elegant molecular machinery we see in every living organism? These questions drive ongoing research into life's origins. Scientists are working to recreate the transition from chemistry to biology in laboratory conditions, studying extremophile organisms that might resemble early life forms, and searching for signs of life on other worlds that might provide clues about how biology emerges from chemistry. Each new discovery reveals both how remarkable life's emergence was and how it might have occurred through natural processes operating over vast spans of time. The birth of everything living represents the universe's greatest creative achievement, the moment when matter learned to organize itself into patterns complex enough to contemplate their own existence. From those first self-replicating molecules in ancient seas to the sophisticated organisms reading these words, life represents an unbroken chain of chemical reactions that have been running continuously for nearly 4 billion years. Every living thing on Earth today carries within its cells the molecular machinery that first emerged in those primordial waters, refined and improved by billions of years of evolution, but fundamentally unchanged in its basic principles. The story of life's birth teaches us that extraordinary complexity can emerge from simple beginnings through the patient operation of natural processes over geological timescales. It shows us that the universe contains within itself the potential for self-organization, creativity, and ultimately consciousness. Most importantly, it reveals that we are not separate from the natural world, but rather represent its most sophisticated expression, matter organized in such a way that it can understand its own origins and contemplate its own future. 2 4/10 billion years ago, something extraordinary happens in Earth's ancient oceans. Tiny blue-green bacteria called cyanobacteria evolve a remarkable new ability. They learn to capture energy directly from sunlight and use it to split water molecules, releasing oxygen as a waste product. This innovation, called photosynthesis, seems like a small improvement in metabolic efficiency. Instead, it triggers the most dramatic transformation in Earth's history, an event so catastrophic that it kills 99% of all living things. Before photosynthesis evolves, Earth's atmosphere contains virtually no oxygen, just 0.03%, compared to today's 21%. The early atmosphere consists mainly of methane, ammonia, and carbon dioxide. Most life forms are anaerobic organisms that derive energy from chemical reactions that don't require oxygen. In fact, oxygen is toxic to these early life forms. It reacts aggressively with their cellular components, destroying the delicate chemistry that keeps them alive. Here's what makes the evolution of photosynthesis so revolutionary. It harnesses the most abundant energy source in the solar system. Every second, the sun bathes Earth with more energy than all human civilization has used throughout history. But this energy arrives as electromagnetic radiation that most chemical reactions can't use directly. Photosynthesis solves this problem by capturing light energy and converting it into chemical energy that living systems can use. The photosynthetic process is extraordinarily complex, requiring dozens of different proteins working in perfect coordination. Light harvesting complexes capture photons and funnel their energy to reaction centers where the actual chemistry occurs. These reaction centers split water molecules with surgical precision, extracting electrons and protons while releasing oxygen gas. The captured energy drives the formation of ATP and other energy-rich molecules that power cellular processes. But photosynthesis does more than just capture energy. It fundamentally changes the chemistry of life itself. For the first time, living organisms can build complex organic molecules directly from simple inorganic compounds. Carbon dioxide from the atmosphere combines with water to form sugars, which then serve as building blocks for all other biological molecules. This process, called carbon fixation, creates the foundation for virtually all life on Earth. As cyanobacteria spread across the oceans, they begin pumping oxygen into the atmosphere at an unprecedented rate. Each molecule of oxygen represents a deadly poison to the anaerobic organisms that dominate early Earth. The rising oxygen levels trigger what scientists call the Great Oxidation Event, the most severe mass extinction in Earth's history. Imagine the scale of this catastrophe. Organisms that have thrived for over 1 billion years suddenly find their environment becoming toxic. The very air becomes poisonous to them. Ocean chemistry changes so dramatically that vast regions become uninhabitable. Ancient ecosystems collapse as the organisms that form their foundation die off in massive numbers. Yet this catastrophe also creates unprecedented opportunities. The few organisms that survive the oxygen crisis inherit a planet with dramatically new possibilities. Oxygen enables much more efficient energy production than any previous metabolic process. Aerobic respiration can extract nearly 20 times more energy from the same nutrients than anaerobic processes. This energy bonanza fuels the evolution of larger, more complex organisms. Some surviving organisms develop sophisticated mechanisms for detoxifying oxygen, or even using it for energy production. Others retreat to oxygen-free environments, deep sediments, volcanic vents, or highly acidic pools, where they persist to this day. The most successful survivors evolve into new types of organisms that not only tolerate oxygen, but require it for their metabolism. The transformation doesn't end there. Around 2 billion years ago, one of the most remarkable events in evolutionary history occurs: endosymbiosis. Larger cells begin engulfing smaller oxygen-using bacteria instead of digesting them. Initially, this might have been simply predation, one organism attempting to eat another, but occasionally the engulfed bacteria survive inside their host cells. This arrangement creates unexpected benefits for both partners. The engulfed bacteria receive protection and nutrients from their hosts. In return, they provide their hosts with the abundant energy produced by aerobic respiration. Over millions of years, these bacterial partners become permanently integrated into their host cells, evolving into the organelles we now call mitochondria. Here's what makes endosymbiosis so extraordinary. It creates organisms with energy production capabilities far beyond anything achieved by earlier life forms. A typical eukaryotic cell can generate 50 to 100 times more energy than a comparable prokaryotic cell. This energy surplus makes possible the evolution of complex internal structures, sophisticated cellular processes, and eventually multicellular organisms. The evidence for endosymbiosis is written into the very structure of our cells. Mitochondria have their own DNA, separate from the DNA in the cell nucleus. This mitochondrial DNA is circular, like bacterial DNA, rather than linear like nuclear DNA. Mitochondria reproduce by dividing, just like bacteria, rather than through the complex process of sexual reproduction used by eukaryotic cells. Most remarkably, mitochondrial DNA is most closely related to the DNA of a group of bacteria called alpha proteobacteria. This genetic evidence strongly suggests that mitochondria evolved from ancient bacterial endosymbionts that took up residence inside larger host cells over 2 billion years ago. A similar endosymbiotic event occurred with photosynthetic bacteria, which became the chloroplasts found in plant cells. Chloroplasts also have their own DNA and reproduce independently, and their genetic material is most closely related to cyanobacteria, the same organisms that triggered the Great Oxidation event. Through endosymbiosis, some eukaryotic cells acquired the ability to perform photosynthesis, eventually giving rise to all plants, algae, and other photosynthetic organisms. The evolution of eukaryotic cells sets the stage for life's next great transformation, the emergence of multicellular organisms. Single-celled eukaryotes possess sophisticated internal organization and abundant energy, but they remain limited by their size. Larger single cells become increasingly difficult to maintain because their volume grows faster than their surface area, making it harder to exchange materials with the environment. Multicellularity solves this problem by allowing organisms to grow large while keeping individual cells small. But multicellular organization requires unprecedented levels of cooperation between cells. Different cells must specialize for different functions, coordinate their activities, and sometimes sacrifice their own reproductive success for the benefit of the whole organism. The evolution of multicellularity represents one of the most profound transitions in life's history. It occurs independently in several different lineages: plants, animals, fungi, and various groups of algae all evolve multicellular organization through different pathways. This convergent evolution suggests that multicellularity provides enormous advantages once the basic prerequisites are in place. The fossil record reveals that multicellular organisms first appear around 1 billion years ago, but they remain relatively simple for hundreds of millions of years. Then, approximately 540 million years ago, something extraordinary happens. In a relatively brief period lasting only 25 million years, evolution unleashes an explosion of creativity that produces most of the major body plans we see in animals today. This event, called the Cambrian explosion, represents one of the most dramatic episodes in evolutionary history. Within this short time span, evolution invents eyes, shells, claws, fins, jointed legs, and complex brains. Organisms develop sophisticated sensory systems, coordinated behaviors, and predator-prey relationships that transform the nature of life on Earth. The Cambrian explosion produces creatures that seem almost alien compared to earlier life forms. Trilobites develop compound eyes with up to 16,000 individual lenses, creating visual systems more sophisticated than many modern animals possess. Anomalocaris grows to over 6 feet in length, becoming the first giant predator in Earth's oceans. Hallucigenia sports defensive spines and walking legs in arrangements so bizarre that paleontologists initially reconstructed it upside down. Here's what makes the Cambrian explosion so remarkable. It demonstrates evolution's extraordinary creativity when new ecological opportunities become available. The evolution of hard shells, complex sensory systems, and predatory behaviors triggers an evolutionary arms race that drives rapid innovation. Prey species evolve better defenses, predators develop more effective hunting strategies, and the resulting feedback loops accelerate evolutionary change. The Cambrian explosion also marks the emergence of complex ecosystems with multiple trophic levels. For the first time in Earth's history, some organisms specialize in hunting other organisms, while others develop sophisticated defenses against predation. This creates complex food webs that support much higher diversity than previous ecosystems, based mainly on bacterial and algal production. But even as life achieves unprecedented complexity and diversity, it faces periodic catastrophes that reset the evolutionary clock. Over the past 540 million years, Earth has experienced 5 major mass extinctions, each one eliminating a large percentage of existing species and fundamentally altering the course of evolution. The most severe of these events occurs approximately 252 million years ago at the end of the Permian period. Massive volcanic eruptions in what is now Siberia pump enormous quantities of carbon dioxide and toxic gases into the atmosphere. Ocean chemistry changes so dramatically that 96% of marine species go extinct. Life on land suffers similar devastation, with over 70% of terrestrial vertebrate species disappearing. The Permian extinction comes closer to ending complex life on Earth than any other event in the fossil record. For millions of years afterward, ecosystems remain impoverished, dominated by a few hardy species that survived the catastrophe. But eventually, life recovers and diversifies again, evolving in new directions that were impossible while the previous dominant groups remained in place. Each mass extinction follows a similar pattern: catastrophic loss of diversity followed by gradual recovery and innovation. The extinction events eliminate established species and disrupt existing ecosystems, but they also create opportunities for surviving groups to diversify and occupy new ecological niches. The most famous mass extinction occurs 66 million years ago, when an asteroid 6 miles in diameter slams into the Yucatan Peninsula. The impact releases energy equivalent to billions of nuclear bombs, triggering tsunamis, wildfires, and a global winter that lasts for years. 75% of all species go extinct, including all non-avian dinosaurs. Here's what makes this extinction particularly significant for our story. It clears the way for mammals to diversify and eventually produce the lineage that leads to humans. For over 150 million years, dinosaurs dominated terrestrial ecosystems, while mammals remained small, mostly nocturnal creatures. The asteroid impact suddenly removes this dominant group, allowing mammals to evolve into larger sizes and occupy ecological niches previously unavailable to them. Within 10 million years of the asteroid impact, mammals diversify into an extraordinary array of forms. Some become large herbivores that fill the ecological roles previously occupied by dinosaurs. Others evolve into predators, aquatic specialists, or flying forms. This mammalian radiation eventually produces primates, and ultimately the human lineage that can contemplate its own evolutionary history. But mass extinctions represent only the most dramatic episodes in life's ongoing transformation. Between these catastrophic events, evolution continuously refines and improves existing organisms while exploring new possibilities. The result is a steady increase in biological complexity and diversity over geological time. Consider the evolution of eyes, one of the most remarkable examples of evolutionary innovation. Eyes evolve independently in dozens of different lineages, producing an extraordinary variety of optical systems. Compound eyes, camera eyes, scanning eyes, and reflecting eyes all represent different solutions to the challenge of gathering visual information about the environment. The human eye, often cited as an example of irreducible complexity, actually evolves through a series of gradual improvements that can be traced in living organisms today. Simple, light-sensitive spots allow organisms to detect the presence or absence of light, Cup-shaped eyes provide directional information about light sources. Adding a lens improves image formation, while complex focusing mechanisms enhance visual acuity. Each step in this progression provides clear survival advantages, demonstrating how complex structures can evolve through natural selection, operating on simpler precursors. The eye's evolution illustrates a general principle. Complex biological systems typically arise through the modification and elaboration of simpler systems rather than through sudden appearance of fully formed structures. The story of photosynthesis, oxygen production, endosymbiosis, multicellularity, and mass extinctions reveals evolution's fundamental character. It's not a ladder leading toward increasing perfection, but rather a branching tree exploring all possible ways of making a living. Each branch represents a different solution to the challenges of survival and reproduction in changing environments. Some solutions prove more successful than others, leading to extensive diversification and long-term survival. Others represent evolutionary dead ends that disappear when environments change or new competitors appear. But every experiment contributes to life's overall exploration of biological possibility space. The transformations we've explored, from the Great Oxidation Event to the Cambrian explosion to the mass extinctions, demonstrate that life's history is characterized by both gradual change and sudden upheavals. Long periods of relative stability are punctuated by brief episodes of rapid transformation that fundamentally alter the trajectory of evolution. These transformations also reveal life's extraordinary resilience. Despite facing catastrophes that eliminate most existing species, life always recovers and often achieves greater complexity and diversity than before. This resilience stems from life's capacity for innovation, its ability to evolve new solutions to environmental challenges through the twin engines of genetic variation and natural selection. Understanding these great transformations provides crucial context for appreciating life's current diversity and predicting its future evolution. The same processes that drove past changes continue operating today, though human activities are now accelerating the pace of environmental change to levels not seen since the major mass extinctions. As we face the possibility of a sixth mass extinction driven by human activities, the lessons from life's past transformations become increasingly relevant. They remind us that while life as a whole is remarkably resilient, individual species and ecosystems can be fragile when faced with rapid environmental change. They also suggest that whatever challenges life faces in the future, the fundamental processes of evolution, variation, selection, and inheritance will continue driving adaptation and innovation. Life's 4-billion-year track record of surviving and thriving through countless challenges provides reason for optimism about its ultimate future, even as we work to minimize the damage from current environmental changes. The great transformations in life's history have brought us to the threshold of an entirely new phase, the emergence of intelligence capable of understanding and potentially directing its own evolution. This development opens possibilities that have never existed before in Earth's history and raises questions that previous generations of organisms could never have contemplated. On April 8, 1960, astronomer Frank Drake arrives at the Green Bank Radio Observatory in West Virginia with an audacious plan. He intends to point the facility's 85-foot radio telescope toward nearby stars and listen for signals from extraterrestrial civilizations. The project, codenamed Ozma after the ruler of the fictional land of Oz, represents humanity's first systematic attempt to contact alien intelligence. Drake begins his search by targeting two nearby sun-like stars, Tau Ceti and Epsilon Eridani, both located within 12 light-years of Earth. For 6 hours each day over several months, he scans these stars at a frequency of 1,420 MHz, a wavelength that travels well through space and might be used by technological civilizations trying to communicate across interstellar distances. The telescope detects only cosmic static and interference from Earth-based sources. No artificial signals emerge from the cosmic background noise. But Drake's experiment launches a scientific revolution that continues today. For the first time in human history, we begin systematically searching for life beyond our planet, using the tools of modern science to address one of the most profound questions imaginable. Are we alone in the universe? Here's what makes this question so compelling. If life emerged on Earth through natural processes, then it should be able to emerge on other worlds with similar conditions. The universe contains over 100 billion galaxies, each containing hundreds of billions of stars. Many of these stars host planetary systems, and some of those planets orbit at distances where liquid water can exist on their surfaces. The numbers are staggering. Conservative estimates suggest our galaxy alone contains at least 100 billion planets. If even a tiny fraction of these worlds develop life, the cosmos should be teeming with biological activity. Yet despite decades of searching, we have found no confirmed evidence of life beyond Earth. This apparent contradiction, known as the Fermi paradox, poses one of the most intriguing puzzles in science. Drake himself quantified this puzzle in 1961, when he developed what became known as the Drake equation. This formula breaks down the probability of detecting extraterrestrial intelligence into several factors: the rate of star formation, the fraction of stars with planets, the resoldon, the fraction of planets where life emerges, the fraction where intelligent life develops, and the length of time technological civilizations persist. When Drake plugged in his estimates for each factor, he calculated that our galaxy might contain between 1,000 and 100 million technological civilizations. If this estimate is correct, then signals from alien cultures should be detectable with current technology. The silence we observe suggests either that life is much rarer than we expect, that intelligence is much less common than we assume, or that technological civilizations destroy themselves before they can communicate across interstellar distances. Recent discoveries have dramatically improved our understanding of several factors in the Drake equation. The Kepler Space Telescope and other planet-hunting missions have revealed that planetary systems are common throughout the galaxy. Nearly every star appears to host at least one planet, and many stars have multiple worlds orbiting them. More importantly, astronomers have discovered thousands of exoplanets orbiting in their star's habitable zone, the range of distances where surface temperatures allow liquid water to exist. These worlds, sometimes called Goldilocks planets, provide the basic conditions that life requires. Water serves as a solvent for biological chemistry, enables the formation of complex organic molecules, and provides a medium where chemical reactions can occur efficiently. But liquid water alone doesn't guarantee habitability. A planet must also have the right atmospheric composition, sufficient atmospheric pressure, and protection from harmful radiation. It needs a stable climate over geological timescales and a supply of essential elements like carbon, nitrogen, and phosphorus. Most challenging of all, it requires the specific combination of conditions that allows simple chemistry to organize itself into living systems. Earth provides a template for understanding these requirements. Our planet orbits at precisely the right distance from the Sun to maintain liquid water on its surface. It has a substantial atmosphere that moderates temperature extremes and protects surface life from harmful solar radiation. Its magnetic field deflects high-energy particles that would otherwise sterilize the surface. Perhaps most importantly, Earth has remained habitable for over 3.5 billion years despite dramatic changes in solar output, atmospheric composition, and surface conditions. This long-term stability appears to result from complex feedback mechanisms that regulate planetary climate processes that might be rare on other worlds. The search for extraterrestrial life has expanded far beyond Drake's original radio surveys. Modern SETI programs monitor millions of stars simultaneously, searching for artificial signals across a wide range of frequencies. Some projects look for optical signals, laser pulses that might be used for interstellar communication. Others search for evidence of large-scale engineering projects that advanced civilizations might construct around their stars. But astronomers have also begun searching for simpler forms of life that might be much more common than technological civilizations. This approach focuses on detecting biosignatures, chemical or physical indicators that living processes are occurring on distant worlds. The most promising biosignatures involve atmospheric chemistry. Life fundamentally alters the chemical composition of planetary atmospheres. On Earth, photosynthesis produces oxygen at levels far above what would exist through purely geological processes. Life also produces gases like methane, ammonia, and various organic compounds that can be detected in planetary atmospheres using spectroscopic analysis. The James Webb Space Telescope represents a quantum leap in our ability to detect these biosignatures. This instrument can analyze the atmospheric composition of exoplanets by studying how starlight changes as planets pass in front of their host stars. Different atmospheric gases absorb specific wavelengths of light, creating characteristic signatures that reveal the planet's chemical composition. Early results from the James Webb Telescope have already detected water vapor in the atmospheres of several exoplanets. One particularly intriguing world, called K2-18b, shows evidence of water vapor and possibly other organic compounds in its atmosphere. Located 120 light-years from Earth, this planet orbits within its star's habitable zone and might possess conditions suitable for life. But the search for life isn't limited to distant exoplanets. Our own solar system contains several worlds that might harbor living organisms. Mars shows compelling evidence of ancient river valleys, lake beds, and ocean basins that suggest the planet once had a much warmer, wetter climate than it does today. Robotic missions to Mars have discovered additional evidence that the planet might once have been habitable, or might even harbor life today. The Curiosity rover found organic compounds in Martian rocks, indicating that the basic building blocks of life exist on the planet. The Perseverance rover is currently collecting samples that might contain fossilized microorganisms from Mars's ancient past. Perhaps most intriguingly, several missions have detected methane in the Martian atmosphere. Methane breaks down rapidly in the presence of ultraviolet radiation, so its continued presence suggests an ongoing source. While geological processes can produce methane, biological activity represents another possible explanation for its presence. Seasonal variations in Martian methane levels add to the mystery. Concentrations spike to 45 parts per billion during certain times of the Martian year, then drop to nearly undetectable levels. This pattern might reflect biological activity that increases during warmer seasons, though alternative explanations involving geological processes remain possible. But Mars isn't the only potentially habitable world in our solar system. Jupiter's moon, Europa, harbors a subsurface ocean that contains more than twice as much water as all Earth's oceans combined. This ocean exists beneath a shell of ice, approximately 15 to 25 kilometers thick, protected from the vacuum of space and heated by tidal forces generated by Jupiter's immense gravitational field. Europa's ocean has likely existed for billions of years, providing the stable conditions that life requires for evolution. The moon's rocky core might provide the chemical nutrients that living organisms need, while hydrothermal vents on the ocean floor could supply the energy sources that drive biological processes. Recent observations suggest that Europa's ocean occasionally breaks through the ice shell, creating geysers that spray water vapor into space. These eruptions provide a potential way to sample the ocean's contents without landing on the surface. Future missions plan to fly through these plumes and analyze them for signs of organic compounds or even living microorganisms. Saturn's moon Enceladus presents similar possibilities. This small icy world also harbors a subsurface ocean heated by tidal forces. Geysers near its south pole spray water vapor and organic compounds into space at velocities of over 800 meters per second. These eruptions create one of Saturn's rings and provide direct evidence of chemical activity in Enceladus's hidden ocean. Analysis of material from Enceladus's geysers reveals a complex mixture of water vapor, organic compounds, and salts that suggests hydrothermal activity on the ocean floor. The Moon's ocean appears to have all the basic ingredients that life requires: liquid water, organic compounds, and energy sources. Some astrobiologists consider Enceladus one of the most promising places to search for life in our solar system. Titan, Saturn's largest moon, presents a completely different type of potentially habitable environment. This world has a thick atmosphere and liquid lakes on its surface, the only other planetary body in our solar system known to have stable surface liquids. But Titan's lakes contain liquid methane and ethane rather than water, raising the possibility that life might evolve using completely different chemistry than Earth life. The discovery of extremophile organisms on Earth has expanded our understanding of where life might exist in the universe. These remarkable creatures thrive in conditions that would be instantly lethal to most life forms. Some bacteria live in acid so concentrated that it dissolves metal, with pH levels reaching -0.6. Others survive radiation doses 1,000 times stronger than levels that would kill humans. Particularly relevant to astrobiology are organisms that thrive in extreme cold, high pressure, and environments with unusual chemistry. These extremophiles demonstrate that life can adapt to conditions very different from those in Earth's moderate surface environments. They suggest that the habitable zone around stars might be much broader than previously thought. Some of the most remarkable extremophiles are hyperthermophiles, organisms that require temperatures above 80 degrees Celsius to survive and can reproduce at temperatures exceeding 121 degrees Celsius. These organisms live around hydrothermal vents and in hot springs, using chemical energy sources that don't depend on sunlight. The existence of hyperthermophiles suggests that life might exist in the subsurface environments of many planetary bodies. Mars, Europa, Enceladus, and other worlds might harbor extensive subsurface biospheres heated by geothermal energy rather than solar radiation. These hidden ecosystems could be much more extensive than any surface life. Recent discoveries have also revealed that life can survive in space itself. Bacteria and other microorganisms can endure the vacuum, radiation, and extreme temperatures of the space environment for extended periods. Some organisms even appear to reproduce more rapidly in microgravity conditions than they do on Earth's surface. These findings support the hypothesis of panspermia, the idea that life might spread between planetary bodies on meteorites and other debris ejected during asteroid impacts. Material blasted off Mars's surface by impact events regularly reaches Earth, and some of this material might carry living organisms, or at least organic compounds that could serve as seeds for life. The search for extraterrestrial life has also been revolutionized by advances in our understanding of life's basic requirements. Scientists have identified the essential elements that all known life requires: carbon, hydrogen, nitrogen, oxygen, phosphorus, and sulfur. These elements are abundant throughout the universe, suggesting that the basic building blocks of life exist wherever planets form. More importantly, researchers have demonstrated that many of life's essential molecules can form spontaneously under a wide range of conditions. Amino acids, nucleotide bases, and other organic compounds have been found in meteorites, comets, and interstellar space. These discoveries suggest that the raw materials for life are distributed throughout the cosmos. Laboratory experiments have shown that simple organic compounds can organize into complex structures, resembling primitive cells, under conditions that might exist on other worlds. These experiments demonstrate that the transition from chemistry to biology might be easier than previously thought, at least for simple forms of life. The accumulating evidence suggests that microbial life might be relatively common in the universe, even if complex multicellular organisms are rare. Simple life forms can survive in extreme environments, use a wide variety of energy sources, and persist for billions of years with minimal environmental change. But detecting microbial life on distant worlds remains extraordinarily challenging. Unlike technological civilizations that might broadcast radio signals, microorganisms leave only subtle traces that are difficult to observe from interstellar distances. Most biosignatures require detailed spectroscopic analysis of planetary atmospheres, observations that are possible only for a small number of nearby exoplanets. The search for life beyond Earth represents more than just scientific curiosity. Finding even simple microorganisms on another world would fundamentally change our understanding of life's cosmic significance. It would demonstrate that the transition from chemistry to biology can occur under different conditions than those that existed on early Earth. More importantly, it would suggest that life represents a general property of the universe, rather than an extraordinarily improbable accident that occurred only once. If life emerges independently on multiple worlds, then the cosmos might be far more biologically active than we currently suspect. The implications extend beyond biology to questions about intelligence, consciousness, and humanity's place in the cosmic story. If simple life is common, then complex life might also emerge frequently given sufficient time and appropriate conditions. This possibility raises profound questions about the nature and distribution of intelligence throughout the universe. As our search capabilities continue improving, we edge closer to answering one of the most fundamental questions in science. Are we alone? Whether the answer is yes or no, it will profoundly impact our understanding of life, intelligence, and our own cosmic significance. The search for extraterrestrial life connects us directly to the broader story of life's evolution on Earth. Every organism that has ever lived on our planet represents a successful solution to the challenges of survival and reproduction. Understanding how life works on Earth provides the foundation for recognizing how it might work elsewhere in the universe. On October 1st, 1990, an international consortium of scientists launches the most ambitious biological project in human history. The Human Genome Project aims to decode the complete genetic instructions that build and operate a human being, all 3.2 billion letters of DNA that distinguish us from every other species on Earth. The scope of this undertaking staggers the imagination. The human genome contains enough genetic information to fill approximately 5,000 books of 300 pages each. If you could read one letter of genetic code per second, it would take you over 100 years to read through a single human genome from beginning to end. Yet this vast library of information fits inside the nucleus of every cell in your body, a space so small that 50,000 nuclei could fit on the head of a pin. Here's what makes the Human Genome Project so revolutionary. For the first time in history, we can read the complete instruction manual that life uses to build complex organisms. Every characteristic you possess, from the color of your eyes to the shape of your brain, emerges from information encoded in this genetic text. Understanding how this code works opens unprecedented possibilities for treating diseases, extending human lifespan, and even modifying the biological processes that define life itself. The project begins with a bold prediction. Despite the enormous complexity of the human genome, scientists believe they can decode the entire sequence within 15 years at a cost of approximately $3 billion. Many experts consider this timeline impossibly optimistic. The fastest DNA sequencing methods available in 1990 can decode only a few hundred letters per day. At this rate, sequencing a complete human genome would require thousands of years. But the Human Genome Project drives rapid advances in sequencing technology. Automated DNA sequencing machines replace slow manual methods. Computer algorithms accelerate the analysis of genetic data. Most importantly, scientists develop new strategies for breaking the genome into manageable pieces that can be sequenced in parallel and then reassembled using sophisticated software. The race to complete the human genome intensifies in 1998 when geneticist Craig Venter announces that his company, Celera Genomics, will sequence the human genome using a revolutionary approach called shotgun sequencing. Instead of methodically working through the genome chromosome by chromosome, Venter's team will randomly shred multiple copies of the genome into millions of small pieces, sequence each piece, and then use computer algorithms to reassemble the complete sequence. This approach requires enormous computational power to analyze the overlapping DNA fragments and identify the correct order for reassembly. But it also allows much faster progress than the systematic approach used by the International Consortium. The competition between these two approaches accelerates research and drives down costs. On June 26, 2000, President Bill Clinton announces that the rough draft of the human genome has been completed. Standing in the East Room of the White House alongside scientists from both competing projects, Clinton declares, "Today, we are learning the language in which God created life. We are gaining ever more awe for the complexity, the beauty, the wonder of God's most divine and sacred gift." The announcement represents a triumph of international scientific cooperation and technological innovation. What many experts thought would take decades has been accomplished in just 13 years. The cost, while substantial at $3.8 billion, proves far less than the original estimates adjusted for inflation. But completing the rough draft is only the beginning. The real work lies in understanding what the genetic code actually means. The human genome contains approximately 20,500 genes, far fewer than scientists expected. Some single-celled organisms have more genes than humans, suggesting that complexity doesn't simply correlate with gene number. Here's what the genome reveals about human nature. We share 99.9% identical DNA with every other human on Earth. The genetic differences between any two individuals amount to only about 3 million letters out of 3.2 billion, a difference of roughly 0.1%. This tiny variation accounts for all the diversity we see in human appearance, physiology, and behavior. Even more surprisingly, humans differ from chimpanzees by only 1.2% of their DNA sequence. We share approximately 98.8% of our genetic code with our closest living relatives. This genetic similarity reflects our recent common ancestry. Humans and chimpanzees diverged from a shared ancestor only about 6 to 7 million years ago. The comparison becomes even more striking when we consider our relationship to other organisms. Humans share about 90% of their genes with mice, 60% with fruit flies, and 26% with baker's yeast. Even bacteria possess many genes that are clearly related to human genes, reflecting the deep evolutionary connections that link all life on Earth. These discoveries fundamentally change our understanding of what makes humans unique. Our distinctive characteristics don't result from having completely different genes than other organisms. Instead, they emerge from subtle differences in how similar genes are regulated and expressed. Small changes in gene regulation can produce dramatic differences in development, physiology, and behavior. The genome also reveals surprising insights about human evolution and migration. By comparing genetic sequences from people around the world, scientists can trace the movements of human populations over tens of thousands of years. The data shows that all non-African populations descend from a small group that left Africa approximately 70 to 80,000 years ago. This genetic evidence indicates that human genetic diversity is actually quite limited compared to other species. The entire human population appears to have passed through a severe bottleneck at some point in our evolutionary history, reducing our species to perhaps as few as 10,000 individuals. All modern humans descend from this small surviving population. But the Human Genome Project represents just the first step in a much larger revolution in biological understanding. Reading the genetic code is one challenge. Editing it is another entirely. In 2012, scientists develop a revolutionary tool called CRISPR-Cas9 that allows precise editing of DNA sequences in living cells. CRISPR, which stands for Clustered Regularly Interspaced Short Palindromic Repeats, originally evolved as an immune system in bacteria. When viruses attack bacterial cells, the CRISPR system captures pieces of viral DNA and stores them as a kind of genetic memory. If the same virus attacks again, the CRISPR system uses this stored information to guide molecular scissors that cut up the viral DNA. Scientists Jennifer Doudna and Emmanuel Charpentier realized that this bacterial immune system could be reprogrammed to cut any DNA sequence. By designing guide RNAs that match specific genetic targets, they could direct the CRISPR molecular scissors to make precise cuts in chromosomes. These cuts allow scientists to remove, replace, or insert genetic material with unprecedented accuracy. Here's what makes CRISPR so revolutionary. It transforms genetic engineering from a slow, expensive, and imprecise process into something that can be accomplished quickly and cheaply in laboratories around the world. Tasks that previously required months or years can now be completed in days or weeks. The cost of genetic modifications has dropped by orders of magnitude. CRISPR applications range from treating genetic diseases to improving crop yields to creating new biomaterials. Scientists have used CRISPR to correct genetic defects that cause sickle cell anemia, muscular dystrophy, and inherited blindness. Agricultural researchers have developed crops with improved nutritional content, better resistance to pests and diseases, and increased tolerance for drought and other environmental stresses. Perhaps most remarkably, CRISPR allows scientists to make changes that could be inherited by future generations. Germline editing, modifying eggs, sperm, or early embryos, could potentially eliminate genetic diseases from family lineages permanently. However, this possibility raises profound ethical questions about the appropriate limits of genetic modification in humans. The power of modern genetic engineering extends beyond editing existing organisms to creating entirely new forms of life. In 2010, Craig Venter's team achieves a milestone that seemed like science fiction just decades earlier. They create the first synthetic bacterial genome and use it to boot up an artificial life form. The project begins with Mycoplasma mycoides, a bacterium with one of the smallest known genomes, only 581,000 genetic letters. Venter's team synthesizes this entire genome from scratch, using laboratory chemicals, then transplants the artificial chromosome into a closely related bacterial cell whose own genome has been removed. The result is a living bacterium whose genetic material is entirely artificial, yet which behaves exactly like its natural counterpart. The synthetic bacterium reproduces normally, suggesting that the artificial genome contains all the information necessary for life. To prove that the organism is indeed artificial, the researchers insert DNA sequences that spell out famous quotations and the names of the scientists involved in the project. This achievement demonstrates that life's fundamental processes can be understood well enough to recreate them artificially. It opens the possibility of designing organisms with entirely new capabilities, bacteria that could produce pharmaceuticals, clean up environmental pollution, or manufacture materials with properties that don't exist in nature. Synthetic biology represents the logical extension of our growing understanding of genetic codes and cellular mechanisms. If life operates according to comprehensible principles, then we should be able to engineer biological systems just as we engineer mechanical or electronic systems. The main difference is that biological systems are self-assembling, self-repairing, and capable of evolution. But perhaps the most profound mystery revealed by genetic research concerns the nature of consciousness itself. The human brain contains approximately 86 billion neurons, each one connecting to roughly 7,000 others through structures called synapses. This creates a network of roughly 600 trillion connections, more synaptic connections in a single brain than there are stars in our galaxy. Here's what makes consciousness so puzzling. Despite this enormous complexity, the brain operates according to the same basic principles as any other biological system. Neurons are cells that follow the same biochemical rules as liver cells, muscle cells, or skin cells. They contain the same genetic information, and use the same molecular machinery for basic cellular processes. Yet somehow, the collective activity of these neural networks gives rise to subjective experience, to the inner world of thoughts, feelings, sensations, and awareness that defines conscious existence. How billions of individual cells, each one following simple biochemical rules, can generate the experience of consciousness remains one of the deepest mysteries in science. Recent advances in neuroscience have begun revealing how specific aspects of consciousness emerge from neural activity. Different regions of the brain specialize in processing different types of information: visual, auditory, emotional, or motor. These specialized regions communicate through complex networks that integrate information and coordinate responses. Researchers have identified neural correlates of specific conscious experiences. When you see the color red, specific patterns of neural activity occur in your visual cortex. When you feel afraid, predictable changes happen in your amygdala and related brain structures. When you make a decision, characteristic activity patterns emerge in your prefrontal cortex. But understanding the neural correlates of consciousness isn't the same as understanding consciousness itself. We can measure the brain activity associated with seeing red, but we still don't understand why this neural activity is accompanied by the subjective experience of redness. This explanatory gap between objective neural activity and subjective experience represents one of the most challenging problems in contemporary science. Some researchers propose that consciousness emerges from the integration of information across different brain regions. According to this view, consciousness arises when the brain creates unified models of sensory information, memories, and predictions about the future. The subjective experience of consciousness might be the brain's way of representing its own information processing activities. Others suggest that consciousness might depend on quantum mechanical processes in the brain. Quantum mechanics allows particles to exist in multiple states simultaneously and enables instantaneous correlations between distant particles. Some theories propose that quantum processes in neural microtubules might give rise to the unified nature of pi, conscious experience. These questions about consciousness connect directly to broader questions about human nature and our place in the biological world. If consciousness emerges from the activity of neural networks following biochemical rules, then understanding these rules might eventually allow us to enhance, modify, or even recreate conscious experience. The implications extend to artificial intelligence and the possibility of creating conscious machines. If consciousness results from specific patterns of information processing, then sufficiently sophisticated computers might eventually achieve conscious awareness. This possibility raises profound questions about the nature of minds, the rights of artificial beings, and the future relationship between biological and artificial intelligence. The genetic revolution has also revealed surprising connections between human biology and the broader story of life on Earth. The human genome contains fossil remnants of ancient viral infections, genetic sequences borrowed from bacteria through horizontal gene transfer, and pseudogenes that reveal our evolutionary history. About 8% of the human genome consists of sequences derived from ancient retroviruses that infected our ancestors millions of years ago. These viral sequences, called endogenous retroviruses, became permanently integrated into our chromosomes and are now passed down from generation to generation like any other genetic material. Most of these viral sequences are inactive, but some have been co-opted for important biological functions. Syncytin, a protein essential for placental development, evolved from an ancient retroviral protein. This means that mammalian pregnancy depends partly on genetic material originally derived from viruses, a remarkable example of how evolution repurposes existing components for new functions. The genome also contains numerous pseudogenes, genetic sequences that resemble functional genes but have been disabled by mutations. These molecular fossils provide evidence of our evolutionary history. Humans have pseudogenes for producing vitamin C, detecting pheromones, and digesting certain plant compounds, capabilities that our ancestors possessed but that we have lost during evolution. Understanding genetic codes has practical applications that extend far beyond basic research. Pharmacogenomics uses genetic information to predict how individuals will respond to different medications. Some people metabolize certain drugs rapidly, while others process them slowly, leading to dramatic differences in drug effectiveness and side effects. Gene therapy aims to treat diseases by modifying genetic information in patients' cells. Scientists have successfully treated inherited immune deficiencies, certain types of blindness, and blood disorders using gene therapy approaches. These treatments work by introducing functional genes into cells that lack them, or by correcting defective genetic sequences. Personalized medicine uses genetic information to tailor treatments to individual patients. Cancer therapy increasingly depends on understanding the specific genetic mutations that drive tumor growth in each patient. Targeted therapies can attack cancer cells with particular genetic characteristics while leaving normal cells unharmed. But genetic technology also raises important ethical questions about privacy, equality, and human enhancement. Genetic information could potentially be used for discrimination in employment, insurance, or social relationships. The ability to modify human genetics raises questions about what types of changes are acceptable and who should make these decisions. As our understanding of genetic codes continues advancing, we approach the possibility of directing our own evolutionary future. For the first time in the 4-billion-year history of life on Earth, organisms can understand and potentially modify the genetic instructions that define their own nature. This capability represents both an enormous opportunity and a profound responsibility. The choices we make about genetic technologies will influence not only our own generation, but potentially all future generations of humans and other organisms. Understanding life's master instructions gives us power that previous generations could never have imagined, and challenges that they never had to face. The story of decoding life's genetic instructions reveals how far we've come in understanding the molecular basis of life itself. From the first glimpses of DNA structure to the complete sequencing of human genomes to the development of precise gene editing tools, each advance has deepened our appreciation for life's extraordinary complexity and opened new possibilities for influencing its future direction. Today, as you read these words, something extraordinary is happening in laboratories around the world. Scientists are discovering that life operates according to principles far stranger and more wonderful than anyone imagined just decades ago. The same quantum mechanical forces that govern subatomic particles also influence the most basic processes of biology. Plants harness quantum coherence to achieve near-perfect efficiency in photosynthesis. Birds navigate using quantum entanglement in specialized cells in their eyes. Some bacteria employ quantum tunneling to speed up the chemical reactions that power their metabolism. These discoveries reveal that the boundary between physics and biology isn't as clear as we once thought. Life doesn't just follow the laws of physics, it exploits them in sophisticated ways that we're only beginning to understand. The quantum realm, once considered the exclusive domain of particle physicists, turns out to be intimately connected to the everyday functioning of living systems. Here's what makes quantum biology so remarkable. Quantum effects typically occur only under extremely controlled conditions in isolated systems at temperatures close to absolute zero, protected from any environmental interference. Yet living systems somehow maintain quantum coherence at body temperature in the wet, noisy environment of cells. This suggests that evolution has discovered ways to harness quantum mechanics that our most advanced technologies haven't yet achieved. Consider photosynthesis, the process that powers virtually all life on Earth. When a photon of sunlight hits a chlorophyll molecule, it excites an electron to a higher energy state. This electron must then travel through a complex network of molecules to reach the reaction center, where its energy can be captured and converted into chemical form. Classical physics predicts that this electron should randomly wander through the molecular network, losing most of its energy as heat before reaching its destination. But experiments reveal that photosynthesis operates with near-perfect efficiency. Almost every absorbed photon contributes to energy production. This efficiency exceeds what should be possible according to classical physics. Recent research shows that the electron exists in a quantum superposition, simultaneously exploring all possible paths through the molecular network until it finds the most efficient route to the reaction center. This quantum effect allows photosynthesis to achieve efficiencies that approach the theoretical maximum allowed by physics. The implications extend far beyond plant biology. Understanding how living systems maintain quantum coherence could revolutionize solar energy technology, quantum computing, and materials science. Nature has apparently solved engineering problems that our most sophisticated technologies still struggle with. Birds provide another striking example of quantum biology in action. Many species navigate across thousands of miles using Earth's magnetic field, detecting magnetic gradients too weak for any known sensory mechanism to perceive. Scientists have discovered that this navigation ability depends on quantum entanglement in cryptochrome proteins found in birds' eyes. When light hits these proteins, it creates pairs of quantum entangled electrons whose spins remain correlated even when separated. Earth's magnetic field influences these quantum states in ways that might allow birds to literally see magnetic field lines as patterns of light and dark in their visual field. This quantum compass provides navigational information accurate enough to guide migrations across entire continents. The discovery of quantum effects in biological systems represents just one frontier in our expanding understanding of life's capabilities. Researchers are also uncovering the mechanisms that control aging, potentially opening pathways to dramatically extending human lifespan. For most of human history, aging seemed inevitable, an unavoidable consequence of wear and tear that accumulates over a lifetime. But recent research reveals that aging is a regulated biological process, controlled by specific genetic programs that can be modified. Studies in laboratory animals have achieved remarkable results. Mice whose lifespans have been extended by 300%, worms that live 10 times longer than normal, and flies that remain youthful far beyond their typical lifespan. These longevity interventions work by targeting fundamental aging mechanisms. Telomeres, the protective caps on chromosomes, normally shorten with each cell division until cells can no longer reproduce. Researchers have developed ways to lengthen telomeres, effectively resetting cellular aging clocks. Other approaches target senescent cells, age cells that stop dividing but continue producing inflammatory compounds that accelerate aging in surrounding tissues. Perhaps most promising are interventions that mimic the effects of caloric restriction, the only proven method for extending lifespan in most organisms. Caloric restriction activates cellular stress response pathways that improve DNA repair, enhance cellular maintenance, and increase resistance to age-related diseases. Drugs that activate these same pathways without requiring dietary restriction could provide the benefits of caloric restriction while allowing normal food intake. The implications of life extension research extend far beyond individual longevity. If aging is a controllable biological process, then many age-related diseases, cancer, cardiovascular disease, neurodegeneration might become preventable or treatable through interventions that maintain youthful cellular function. But extending human lifespan raises profound questions about the future of human society. How would dramatically longer lifespans affect economic systems based on retirement and generational transfer of wealth? What would it mean for population growth and resource consumption? How would extended lifespans change human relationships, career paths, and life goals? These questions become more pressing as artificial intelligence begins augmenting and potentially surpassing human cognitive capabilities. Machine learning systems can already recognize patterns in genetic data that human scientists miss, design new proteins with desired properties, and accelerate drug discovery by orders of magnitude. The partnership between human intelligence and artificial intelligence is transforming biological research. AI systems analyze genomic data from millions of individuals to identify genetic variants associated with diseases. They predict protein structures from amino acid sequences, solving problems that have challenged biochemists for decades. They design new enzymes, antibodies, and other biological molecules with properties that don't exist in nature. But AI is also revealing new aspects of intelligence itself. Large language models can engage in conversations that seem remarkably human-like, yet they operate using principles completely different from biological intelligence. They process information in parallel rather than sequentially, learn from vast data sets rather than direct experience, and make decisions using statistical patterns rather than conscious reasoning. This divergence between artificial and biological intelligence raises fundamental questions about the nature of consciousness, creativity, and understanding. If machines can perform intellectual tasks that we associate with consciousness without actually being conscious, what does this tell us about the nature of mind itself? Meanwhile, the search for life beyond Earth has entered a new phase with the deployment of increasingly sophisticated detection methods. The James Webb Space Telescope can analyze the atmospheric composition of exoplanets by studying how starlight changes as planets pass in front of their host stars. Early observations have already detected water vapor in the atmospheres of several distant worlds. One particularly intriguing discovery involves K2-18b, a planet located 120 light-years from Earth. This world orbits within its star's habitable zone and shows spectroscopic evidence of water vapor, carbon dioxide, and possibly other organic compounds in its atmosphere. While these observations don't prove the existence of life, they demonstrate that the basic conditions for biology exist on worlds far from our solar system. The detection of biosignatures in exoplanet atmospheres represents one of the most promising approaches for finding extraterrestrial life. Oxygen, methane, and other gases produced by biological processes create distinctive spectroscopic signatures that can be detected across interstellar distances. The challenge lies in distinguishing biological production of these gases from geological processes that can produce similar atmospheric compositions. But the search for life isn't limited to distant exoplanets. Missions to Mars continue, uncovering evidence that the planet might harbor life today or might have hosted life in the past. The Perseverance rover is collecting rock samples that might contain fossilized microorganisms from Mars's ancient past, while other missions search for signs of current biological activity. Perhaps most intriguingly, robotic missions to Jupiter's moon Europa and Saturn's moon Enceladus could sample the subsurface oceans that exist beneath these worlds' icy surfaces. These oceans contain more water than all Earth's oceans combined, and might harbor ecosystems based on chemosynthesis rather than photosynthesis. The possibility of finding life in our own solar system has profound implications for understanding life's cosmic significance. If life emerges independently on multiple worlds within a single planetary system, it suggests that biological processes might be far more common throughout the universe than we currently suspect. But even as we search for life elsewhere, we face the reality that life on Earth is experiencing its sixth mass extinction. Species are disappearing at rates 1,000 times faster than natural background extinction rates. Climate change, habitat destruction, pollution, and other human activities are driving biodiversity loss that rivals the great extinction events of the geological past. This crisis reflects the extraordinary impact that one species, humans, can have on planetary systems. In just a few centuries, human activities have altered atmospheric composition, changed global climate patterns, and triggered ecological changes that affect every ecosystem on Earth. We have become a geological force comparable to volcanic eruptions or asteroid impacts, yet the same capabilities that allow humans to impact planetary systems also provide tools for addressing these challenges. Synthetic biology could create organisms that remove carbon dioxide from the atmosphere, break down plastic pollution, or restore damaged ecosystems. Gene drives could control invasive species or disease vectors. Precision agriculture could feed growing populations while reducing environmental impacts. The key challenge lies in developing these technologies responsibly while addressing the ethical and social implications of human enhancement, environmental modification, and potential contact with extraterrestrial life. We stand at a unique moment in Earth's history, the first time that a species has become aware of its own evolutionary origins and gained the power to direct its own future development. This awareness brings both unprecedented opportunities and enormous responsibilities. The choices we make about genetic technologies, artificial intelligence, environmental management, and space exploration will influence not only our own species, but potentially all life on Earth and beyond. The questions that first stirred in ancient tidal pools have evolved into challenges that will define the next phase of life's cosmic story. How we answer these questions will determine whether intelligence represents a permanent feature of the universe or merely a brief experiment in cosmic evolution. As we stand on the threshold of potentially revolutionary advances in longevity, intelligence enhancement, and space colonization, we carry with us the accumulated wisdom of 4 billion years of evolution. Every cell in our bodies contains molecular machinery refined by countless generations of natural selection. Every thought in our minds reflects the operation of neural networks shaped by millions of years of cognitive evolution. Yet we also possess capabilities that transcend anything achieved by previous life forms. We can understand our own genetic codes, modify our own biological processes, and potentially spread life beyond the planet where it originated. We can contemplate abstract concepts, create works of art, and ask questions about the meaning and purpose of existence itself. This combination of evolutionary heritage and transcendent capability places us in a unique position in the cosmic story. We are simultaneously products of natural selection and agents capable of directing future evolution. We are both biological organisms, subject to the same physical laws as bacteria, and conscious beings capable of contemplating the nature of consciousness itself. The endless frontiers that stretch before us include not only technological and scientific advances, but also fundamental questions about what it means to be alive, intelligent, and conscious in a universe that seems indifferent to our existence, yet has somehow given rise to beings capable of understanding their own nature. Perhaps the most remarkable aspect of life's story is that it continues unfolding. Every day brings new discoveries about the mechanisms that govern living systems, the potential for life elsewhere in the universe, and the possibilities for enhancing and extending human capabilities. The questions that drive this research connect us directly to the curiosity that has motivated human inquiry since our species first wondered about the nature of life itself. As we peer deeper into the quantum realm, reach farther into space, and develop more sophisticated tools for understanding and modifying biological systems, we participate in the same process of exploration and discovery that began when the first humans looked at the natural world and asked, "How does this work?" The answers we discover will shape not only our understanding of life's past, but also our ability to influence its future. In this ongoing quest to understand life's endless frontiers, we continue the conversation that began in primordial seas, and that will likely continue as long as conscious beings exist to ask questions about their own existence. The story of life on Earth teaches us that the universe contains within itself the potential for extraordinary creativity, complexity, and beauty. From simple molecules learning to copy themselves to conscious minds capable of contemplating infinity, life represents the universe's greatest experiment in self-organization and self-awareness. That experiment continues today in laboratories and observatories around the world, in the depths of Earth's oceans and the vacuum of space, In the quantum realm of subatomic particles and the vast scales of cosmic evolution, each new discovery adds another chapter to life's ongoing story, a story that we are uniquely privileged to read, understand, and help write.